Hi, everyone. Welcome to Creative Biolabs live webinar series. To start, I would like to briefly introduce Creative Biolabs to all our attendees today. So Creative Biolabs leverages advanced nucleic acid technology platforms and a team of experienced scientists to propel the development of mRNA technology, tackling the distinct challenges associated with mRNA drug development. Our expertise encompasses a wide range of areas, including mRNA biology, chemistry formulation and delivery, bioinformatics, and protein engineering. Today's webinar is titled EABR Nanoparticles as a Platform Technology for Hybrid mRNA Vaccine Development. Recent scientific advancements have transformed vaccine development through mRNA technology, particularly in the fight against infectious diseases such as COVID-19, and mRNA vaccines function by instructing cells to produce a protein that elicits an immune response, effectively training the body to recognize and combat the virus. This innovative approach not only allows for faster production times, but also offers adaptability to emerging variants, making it an essential tool in modern medicine's fight against pandemics. In this session, we are excited to have Dr. Magnus Hoffmann guiding us through the application of hybrid mRNA technology in vaccine development, strategies for optimizing hybrid mRNA vaccines, and its potential in creating pan-coronavirus vaccines. Now let me introduce our speaker for today, Dr. Hoffmann. He is an assistant investigator at the Gladstone Institute of Virology and the Gladstone Institute of Data Science and Biotechnology. Before moving to Gladstone, he was the inaugural Merkin Institute Fellow at the Merkin Institute for Translational Research at the California Institute of Technology. Based on his graduate work in Pamela Bjorkman's laboratory at Caltech, he received the Milton and Francis Clauser Prize for the best PhD thesis across all disciplines, and he was awarded an NIH Director's Early Independence Award to launch his own laboratory as an independent postdoctoral scholar at Caltech. Dr. Hoffmann's research focuses on the development of innovative vaccine technologies and gaining a deeper understanding of the immunological mechanisms that shape vaccine-induced immune responses. He developed the EABR technology, an innovative approach to genetically encode nanoparticles for vaccine applications. This vaccine platform combines features of mRNA and protein nanoparticle-based vaccines, resulting in superior neutralizing antibody responses against original and variant SARS-CoV-2 in mice. Now, ongoing and future research in his group focuses on the continued optimization, evaluation, and application of this technology. Now, before Dr. Hoffmann begins his presentation, I want to quickly remind our audience that we will have a Q&A session at the end of the presentation. If you have any questions during the presentation, please type your questions into the Q&A panel, and then Dr. Hoffmann will get to them after his talk is finished. Now, let's welcome Dr. Magnus Hoffmann. And Dr. Hoffmann, thank you so much for presenting your important findings for us to us today. And you may begin when ready.
Okay, great. Thank you so much. Let me share my slides. Okay, can you see that? I'll go on the slideshow. Yes. Yeah. Okay.
Yeah.
All there?
Perfect.
Okay, great.
Yes.
Wonderful.
Wonderful.
Well, thank you so much again for the wonderful introduction, and it's really a great pleasure to be here today and share my research with you all. So as was mentioned in the introduction, my talk will focus today on the EABR nanoparticle technology that I developed as a graduate student in Pamela Bjorkman's lab at Caltech. And then I would like to discuss how we're using this technology to develop what we call hybrid mRNA vaccines against various infectious diseases. And we call them hybrid mRNA vaccines because they combine attributes of both mRNA vaccines as well as protein nanoparticle-based vaccines. And so before I start, I would just like to disclose that I am a paid consultant and have equity interest in a company called Vaccine Company. Vaccine Company develops vaccines against various infectious diseases and may potentially benefit financially from the research findings presented here. Of course, you know, we are at the tail end of a global devastating pandemic. And so it's not going to be that hard for me to convince you all that innovative vaccine technologies and future pandemic preparedness are really important. But nevertheless, I was interested to see what people think. So I came across this survey here from the UK, where they asked people, which of the following, if any, do you think are the most likely to cause human extinction? And so, as you can see, pandemics are actually like soundly in third place right after climate change and nuclear war, which I think is acceptable. And good news is they are much higher place than, let's say, a zombie outbreak, which I think is reassuring. So clearly pandemics are a scary thing, and people think that this could be a threat to human extinction even. The interesting thing about the survey was that this took place in 2016. So that's actually before the COVID-19 pandemic. So it would be really interesting to see what the results would look like today. I actually don't know in which direction it would go. It could either be that people say like, oh, wow, we've seen this now. A pandemic can be absolutely devastating. And so we think it's higher place, but it could also be lower place because they'll be like, well, okay, it was devastating, but we weren't close to human extinction. So, I don't know how people would think about it, but I think it's interesting. And of course, humanity has faced numerous devastating pandemics, over the last couple of centuries. And so this is a timeline here, and each of these spheres represents one of the pandemic outbreaks. And so the larger the sphere, the more lives, the more people were killed during the outbreak. And so you can see, for instance, here we have the Black Death, that cost 200 million lives in the 14th century. Then, of course, there was the smallpox outbreak. We have the Spanish flu in more recent times. This was 1918, where 40 to 50 million people lost their lives. And then, of course, we have the still ongoing HIV/AIDS pandemic. So what's striking is that in the last 20 years, the three of the outbreaks that we detected were actually caused by beta coronaviruses. So this is namely SARS, MERS, and COVID-19. Of course, the SARS and MERS caused relatively small outbreaks, which we were lucky about because they were really lethal. But these events still highlight that future pandemic coronavirus outbreaks are a major global health concern. And of course, vaccines are a very important strategy for us to protect ourselves against future coronavirus outbreaks, but of course also outbreaks with other viruses. And there are really two key vaccine strategies that are important. And so the first one is rapid response vaccines. So this is exactly what we saw during the COVID-19 pandemic. You detect a new outbreak caused by a new virus, and then you sequence that virus, and then you develop a vaccine in response to the outbreak. And so, of course, the absolute key here is that you need technologies that enable rapid development in large-scale manufacturing of effective vaccines. The second vaccine strategy would be to make universal coronavirus vaccines. And of course, the goal here would be to elicit broad immunity to a wide range of coronavirus strains before human transmission has even occurred. So this is more a preventive strategy. And of course, to achieve that, you need vaccine strategies that target immune responses to parts of the virus that are highly conserved across the family of coronaviruses, so that you can get protection against a lot of different strains, because you don't know which one is going to be the one that is going to infect you. So my lab is actually very focused on both of these vaccine approaches, and I will discuss them both today. I will start with our work on making more effective rapid response vaccines. Obviously, as you all know, the COVID-19 mRNA vaccines were developed in record time. The most remarkable part for me is that it only took them 2 months from January 12 to March 16, from the day that the SARS-CoV-2 genomic sequence was published to the day when the first human participant was immunized with an mRNA vaccine in a phase 1 clinical trial. So that's like really fascinating that they could do that so fast. And this is really one of the strengths of the mRNA vaccine technology. You simply just cut and paste the new sequence and you start the manufacturing process. Other types of vaccines, such as protein subunit-based vaccines or attenuated virus-based vaccines, take a lot longer to develop. And now, of course, the adenovirus-based vaccines were also developed very fast, but they weren't quite as effective as the mRNA vaccines, and there were also more health concerns. So that's why mRNA vaccines are really superior technology when it comes to future pandemic preparedness. So they are an amazing technology, but they do have some problems. So the first problem is that COVID-19 mRNA vaccine-induced antibody responses contract pretty rapidly over time. This is data here from individuals that either received the initial prime boost vaccinations with the Moderna shot on the left or the Pfizer-BioNTech shot on the right. And so you can see here that the peak antibody titers were measured on day 42. So this is 2 or 3 weeks after the second dose, depending on which shot you received. And you can then see that on day 185, so this is about 4.5 months after the second immunization, you can see that the antibody titers already dropped by over 5-fold. And some of the individuals at this time point might not be fully protected anymore. And so this is obviously a problem. And then the second problem is that viruses such as SARS-CoV-2 obviously mutate pretty rapidly, and they can basically acquire escape mutations that allow them to evade from immune responses that were elicited either by vaccination or prior infections. And so this is data here showing that the mRNA vaccine-induced antibodies neutralized the original variant, Wuhan variant, very effectively. And they were still pretty effective against the Alpha, Delta, and even to some degree the Beta variant. But you can see that the activity almost completely drops against the Omicron variant, which obviously acquired a lot of mutations in the spike protein that enable very effective immune escape. And so because of this limited durability of the antibody responses and the relative ineffectiveness against emerging viral variants, current COVID-19 mRNA vaccines require frequent and updated booster shots. And so there is a great need for innovative vaccine technologies that can induce more lasting protection in the face of a pandemic. And so, of course, if we want to make the mRNA vaccines more effective, we need to understand how they work. So this schematic shows how a regular COVID-19 mRNA vaccine works. So you start off with an mRNA shown here that encodes the SARS-CoV-2 spike protein, which is the main target of our antibody responses. So the mRNA then gets packaged in these lipid nanoparticles, or LNPs, which then effectively deliver the mRNA into our cells. And then inside the cells, the mRNA gets translated into spike protein. And then two things happen. The spike protein gets presented on the surface of the cell. So it's not secreted like some people think. It's actually presented on the cell surface. And that activates B cells, which are obviously the cells that produce our antibodies. And the spike protein also gets chopped up into little peptides that then get presented on MHC molecules, which activates T cells. And so if you think about it, an mRNA vaccine really kind of mimics an infected cell. So this is kind of what an infected cell would look like during an infection, and that can signal the immune system, hey, there's an infection going on, please develop a good response against this virus. And so this is good, but we know there are also other vaccine approaches, such as the Novavax vaccine, which some of you guys may have gotten a shot from, which presents dense arrays of the spike protein on the surface of virus-like nanoparticles, or VLPs. And so these type of vaccines essentially mimic the virus itself, right, to activate immune responses. And they're also very good at activating B cell responses, not as good as mRNA vaccines in terms of activating T cell responses, but also very effective at activating the B cells. And so what we're trying to do is to develop a hybrid mRNA vaccine technology that combines both of these approaches into one. And so the way this looks like is it actually looks very similar to a regular mRNA vaccine, but we engineer the spike protein here on the cytoplasmic tail so that when it gets to the cell surface, it induces the self-assembly and budding of these virus-like nanoparticles that can then biodistribute inside the body and activate particularly B cells more effectively. And so how do we accomplish that? So there's actually a very important pathway in all our cells, which is called the escort pathway. And so escort proteins are really important for facilitating membrane remodeling and scission events inside the cells. And so they're very important in various different cellular processes, such as cell division or exosome biogenesis. And a lot of envelope viruses, including HIV, for instance, actually hijack this pathway during the viral budding process. And so that's exactly what we're doing as well. So when I was a graduate student in Pamela Björkman's lab at Caltech, I developed a technology which we call the EABR technology. And so the way it works is you have your protein of interest. So in our case, this is the SARS-CoV-2 spike protein. And then you insert a short peptide, it's only about 60 amino acids long, at the end of the cytoplasmic tail, here shown in green. And so we call this peptide the escort and Alex binding region, or short EABR. And so this EABR peptide then recruits proteins from the escort machinery, and that induces the self-assembly and budding of these VLPs. And so making these VLPs is actually very simple. So you first have to clone your construct. So in this case, it would be the SARS-CoV-2 spike protein, and then you just insert this EABR peptide at the end, at the C terminus, and then you transfect this construct into, let's say, XB293 cells, but it also works in other cells. And then you can harvest the supernatants of this culture after 48 to 72 hours. And then you can purify these VLPs from the supernatants by ultracentrifugation on a sucrose cushion. And so this is what these purified VLPs look like. Is the movie playing? Yeah, it's right. Okay, great. So this is cryo-electron tomography, and you can see in the red boxes we have these VLPs basically showing up, and it's basically like zooming through them. And so if we zoom in on some of these particles, you can see that the scale bar is 30 nanometers, so the particles are about 40 to 60 nanometers in diameter. And you can see that they are surrounded by a lipid bilayer, which we essentially store from the plasma membrane during the budding process. And then you can see that they're densely coated with these spike proteins on the surface. And based on these tomograms, we can then model what an entire VLP might look like, as you can see here on the right. And so you can see that a single particle might have somewhere between 20 to 40 spikes on the surface, which is actually a higher density than you would see on the real SARS-CoV-2 virus, simply because our particle is actually smaller than the real virus, which is more around like 100 nanometers. Okay. The EABR technology actually has also a second component, which we call the endocytosis preventing motif, or short EPM. And so the EPM was derived from the mouse Fc gamma receptor 2b. And so the way we think the EPM works is that it tethers the protein to the cytoskeleton, and that prevents co-localization or localization into clathrin-coated pits, and thereby that prevents endocytosis of your protein of interest. And so we hypothesize that inserting this EPM peptide, again, it's about 50 amino acids long. If you add that upstream of the green EABR peptide, you could further enhance overall expression of the spike protein, and that could then also result in improved virus-like particle budding. And so we tested this here on the right, where we had two constructs. On the left, it's a spike EABR construct without the EPM. So I understand the nomenclature is a little confusing here. So it basically has no EPM, while the spike EABR construct on the right actually has the EPM, because we always like included that as our default construct. And so we basically transfected these two constructs side by side into XB293 cells, then purified the VLPs from the supernatant, and then ran a Western blot on the purified VLP fraction to see how much spike protein is there. And you can clearly see that adding the EPM increases how much spike protein is in the purified VLP fraction. So now, of course, based on this data, it's impossible to say whether we actually increased how many VLPs we made, or whether we increased how much spike protein is presented per VLP. And so, of course, we still need to do some more experiments here, but overall, it does look like the EPM is enhancing this effect, which is great. And so the next, we wanted to compare the production efficiency of these spike EABR VLPs compared to other approaches that you could use to genetically encode virus-like particles for vaccine applications. And so we tested this here against a couple of other conditions. So on the left, you just had spike protein by itself, which is just a negative control. Obviously, it wouldn't make any VLPs. Then the second condition, you have spike together with the HIV gag protein. So HIV gag is the key structural protein for HIV that forms the capsid and the matrix and the nucleocapsid. And so the GAG expression in a cell just triggers VLP production. And so if you then co-express spike, the idea would be that spike would localize to the surface of the VLPs. And so if you co-express them together, you should make VLPs that have spike on the surface. The third approach was to co-express spike together with the SARS-CoV-2 structural proteins M, N, and E. So again, literature has shown that co-expression of these 4 proteins results in the production of virus-like particles for SARS-CoV-2. And then the final comparison that we included was a spike ferritin fusion protein, where basically ferritin forms these protein capsids, and then spike gets presented on the surface. And so we basically transfected them all side by side, purified the VLPs, and then again ran a Western blot to see how much spike protein is there. And as you can see, at a sample dilution of 1 to 200, the spike EABR, you can only see a very intense band for the spike EABR sample, which shows that it is a more effective way to make VLPs than these other approaches. But it's not like the other approaches weren't there at all. So if you do a sample dilution of 1 to 20, you can see at least bands for spike plus GAG or the spike ferritin construct. But overall, this data shows that our EABR technology is a very effective way to make virus-like particles for vaccine applications. But of course, so it works really well for SARS-CoV-2 spike. But of course, we want to know, like, does this also work for other proteins? And so we tested this here for 2 other proteins. On the left, you see this for a HIV-ONV protein, which of course is also a very important vaccine target for HIV vaccine development. And you can again see here that an ONV-EABR construct produces a lot more virus-like particles than, let's say, co-expression of ONV and GAG. And this is HIV GAG, so this is like sort of a very important comparison, and it's the EABR is clearly better here. And then on the right side, we also tested this for the human CCR5 protein, which is actually a G protein coupled receptor. So a completely different architecture of a membrane protein with 7 transmembrane domains. And you can see that, again, CCR5 EABR works much better than co-expression of CCR5 and GAG, which is great. Okay, so next we then wanted to see whether our SARS-CoV-2 spike EABR VLPs are actually immunogenic in vivo, right? And so we performed a mouse experiment where we immunized the mice twice on day 0 and day 28. And we compared the following 3 vaccine conditions. So in gray, you have the soluble spike protein, which is just like a control. We know this is not going to be very immunogenic. Then in blue, we have spike protein conjugated to spike catcher MI3 nanoparticles, which is another alternative vaccine platform. And we know that these particles work extremely well. So they're like really like kind of like a benchmark in this experiment to see if our particles can be similarly immunogenic as these spike MI3 nanoparticles. And then in red, you have our spike EABR VLPs. And we gave all of these different vaccines with the same adjuvant. In this case, it was the Sigma adjuvant system. And then after the immunizations, we took serum from the animals to measure neutralizing antibody titers. And so you can see here on day 42, so this is 2 weeks after the second immunization, you can see that the spike EABR VLPs induced about 20-fold higher neutralizing antibody titers compared to the soluble spike protein. And the antibody titers were also comparable to the spike MI3 nanoparticles. So that shows that our technology is really good and that these VLPs are highly immunogenic and work well in vivo. So this is good news. But of course, what we were really interested in is, can we use this technology to develop a hybrid mRNA vaccine technology that basically encodes these self-assembling EABR VLPs to combine attributes of both an mRNA vaccine and a protein nanoparticle-based vaccine? And so to test this, we performed another mouse experiment where, again, we immunized the mice twice on day 0 and day 28. And here we compared the following 3 conditions. So now in gray, you have our spike EABR VLPs together with an adjuvant. So these are protein-based purified VLPs that we made in the lab, mixed with an adjuvant, and directly injected into the animals, okay? So this is kind of like a Novavax vaccine, so very similar to that. In blue, you have a regular spike encoding mRNA vaccine. So this is very similar to the Pfizer or Moderna shots. And then in red, you have our hybrid spike EABR mRNA vaccine, where basically the mRNA encodes these self-assembling spike EABR VLPs. And so then again, we looked at the neutralizing antibody responses against SARS-CoV-2, and you can see that on day 56 here on the left, our hybrid spike EABR mRNA vaccine approach actually elicited about 5-fold higher neutralizing antibody titers compared to both the purified VLPs in gray and the regular spike encoding mRNA vaccine in blue. And of course, this was a really exciting result for us. And importantly, on day 112, so this is about 3 months after the second immunization, these differences in antibody titers were maintained, which might suggest that our hybrid mRNA vaccine strategy could also induce more durable antibody responses. But of course, mice it's a difficult animal model to assess that because the antibody titers don't quite drop as fast as they do in humans. So we need to do some more work there. But of course, we also want to test these antibody titers against some of the variants of concern. So we tested this here against the Delta variant on the left, and also the early Omicron-based variants, BA1 and BA2 in the middle and on the right. And what you can see is that our spike EABR hybrid mRNA vaccine approach in red induced a 5 to 15-fold higher neutralizing antibody titers against all of these variants. And this was particularly cool for us to see because it requires at least 3 immunizations with a regular mRNA vaccine to get robust titers against Omicron-based variants in animal models. And so it was really cool for us to see that we were able to already achieve that with 2 immunizations with our hybrid mRNA vaccine technology, which I think is really promising. Okay, so at the end of the study, we then also want to look at T-cell responses. And so we isolated the splenocytes from the immunized animals and then stimulated them with peptides derived from the SARS-CoV-2 spike protein. And then we performed ELISpot assays to measure the secretion of 2 cytokines. We have interferon gamma on the left and IL-4 on the right. And so what you can see is for the interferon gamma secretion, both of the mRNA vaccines, the regular as well as the hybrid mRNA vaccines in blue and in red, induced very potent responses, which were much more potent than the responses induced by the purified VLPs. And this is to be expected. This is consistent with what we've seen in the literature. And so obviously for interferon gamma, it's very hard to say whether the interferon gamma secretion came from CD8 T cells or Th1 CD4 T cells. But overall, it looks like that the hybrid mRNA vaccine strategy maintains the ability to induce potent cytotoxic T cell responses. In terms of IL-4 secretion on the right, you can then see that our hybrid mRNA vaccine approach induced stronger responses than both the purified VLPs as well as the regular mRNA vaccine. And that, of course, is also consistent with the improved antibody titers that we saw. So overall, we still need to do a lot more analysis on the T-cell responses, use more flow cytometry-based assays. But based on the ELISpot results, it looks like our approach also induces very potent T-cell responses. Okay, so in conclusions for this part, the EABR VLP self-assembly is induced by escort recruitment to the cytoplasmic tail of membrane proteins, and we have already demonstrated this to work well for various different coronavirus spikes, as well as other immunogens such as HIV-ONV or influenza hemagglutinin. EABR nanoparticles can be genetically encoded to then develop what we call hybrid mRNA vaccines. And these hybrid mRNA vaccines could solve 2 of the biggest problems with current COVID-19 vaccines, which is they could induce more durable immune protection and also more effective protection against viral variants. And that together could then reduce the need for frequent and updated booster shots. Okay, so obviously you can see this, it's an exciting new approach, and there's a lot of ongoing work on future directions that we're focusing on at the moment. And so the key terms here are really evaluation, optimization, characterization, and application. And so I will not go through all of these parts, but I just want to share with you a little bit what we're doing at the moment. So in terms of evaluation, we really wanted to see if we can apply our hybrid mRNA vaccine platform to develop more effective booster shots for SARS-CoV-2. And so, of course, the initial experiment was performed in naive mice. But of course, that doesn't really reflect well what the human population looks like now, because of course, most of us, if not all, have already received multiple vaccinations, and we probably already been infected a couple of times as well. So we are pre-immunized. And so we wanted to design a mouse study that kind of mimics that a little bit better. And so we performed the following experiment here. We had 4 groups of 10 mice each, and all of the groups initially received the same prime boost regimen. So all of them received a Washington 1 spike mRNA vaccine, which is very similar to the Pfizer Moderna shots. And so they received them on day 0 and day 28. And then we took the serum sample on day 56 and measured the neutralizing antibody responses against the Omicron variant BA.5, which circulated a while ago. And so then you can see on day 56, all of the 4 groups, I mean, they all received the same prime boost, all had pretty low but pretty consistent neutralizing antibody titers across the groups. And so then we waited for a long time. We actually waited for 6 months until day 230. And then we gave each of the 4 groups a different booster shot. And so group 1 received a 3rd dose of the Washington 1 spike mRNA vaccine. And this is actually what happened in real life. You remember all of us actually received a 3rd shot with the same vaccine that we had initially received. And so you can see that on day 244, the neutralizing antibody titer against BA.5 increased about 3-fold for group 1 compared to day 56. Okay, so you got about a 3-fold increase by getting that 3rd shot for group 1 in light blue. This group 2 received a Washington 1 spike EABR mRNA booster. So here we just added the EABR sequence, and you can see that in red on day 244, the antibody response against BA.5 was actually 16-fold higher than on day 56. So that is about a 5-fold higher increase than we saw for group 1. So that was very exciting for us to see. And then in dark blue, group 3 actually received a bivalent Washington 1 BA.5 spike mRNA booster. So this is what in real life we all received as the 4th shot, right? That was like the first bivalent shot that we received. And so you can see groups 2 and 3 actually had the same titer on day 244, which was exciting for us because that shows that the EABR technology might reduce your need to update the booster shots that regularly. So that's an exciting result. And then finally, group 4 received a bivalent Washington 1 BA.5 spike EABR mRNA booster. And so in this group also induced the highest neutralizing antibody titers against BA.5, which was about 2.5-fold higher than the groups 2 and 3, and over 10-fold higher than group 1. And so this was exciting data for us because it shows that the EABR technology can also be very effective when applied as a booster shot for SARS-CoV-2. And of course, we're still in the process of analyzing this data against more strains, et cetera, and hopefully we can publish another manuscript soon. In terms of optimization, I do want to share some ongoing work with you guys where we're trying to design improved EABR sequences. So despite all of these, very promising responses, the original EABR sequence actually has a flaw. And so this is data from a very simple in vitro transfection experiment in HEK293 T cells. And so we're comparing 3 different conditions. So in gray, you have just untransfected control cells. And then in red, we transfected the cells with an mRNA encoding a HIV-ONV immunogen. So this is, just a different immunogen, but similar concept as for SARS-CoV-2 spike. So it's just HIV-ONV, and it's just a cell surface immunogen. And then in orange, you have an ONV EABR construct. So this one has been engineered so it can make the virus-like particles. And so then we measured the expression of the immunogen in 2 different ways. So on the left side, we're looking at cell surface expression. So we basically just take the transfected cells and perform flow cytometry. And then on the right side, we're measuring the VLP production rate. So here we're taking the supernatants from the transfected cells, purify the VLPs, and then run an ELISA to see how much of the immunogen is in that purified VLP fraction. And so what you can see is in red for the standard ONF immunogen, we get very robust cell surface expression. And of course, we don't see any VOPs being produced, which we wouldn't expect because they're not engineered to produce VOPs. So that makes sense. But then if we look at the ONF EABR construct, we do see some VOP production. It's not quite as impressive as for SARS-CoV-2 spike. ONF is a little tricky. It doesn't express quite as well as spikes, but we do see some VOP production here. But then when we look at the cell surface expression, it's like basically gone. We see this massive reduction in cell surface expression. And so initially we weren't quite sure what causes this reduction. We hypothesized it might be because the ONF proteins get incorporated into the budding VOPs and therefore disappear from the cell surface. And, that would have been perfectly plausible. But it turns out, as we were doing more and more experiments, that it seems like that the addition of the EABR sequence actually reduces the overall expression of the immunogen. And of course, that's something that we don't want. We want as much expression as possible so that the vaccine can be as effective as possible. And so we spend a lot of time designing libraries of different new escort recruiting domains to see if we can find a sequence that doesn't have this problem. And I just want to share with you some data on one of the most promising constructs that we have right now. So we call this construct P6V5.1 because it has been derived from the HIV P6 protein, which is the part of the HIV gag protein that recruits the escort machinery during HIV budding. And so what you can see here in blue is that the cell surface expression is actually almost identical to the cell surface expression we see for the unmodified ONF immunogen. And this increase in expression also then results in higher virus-like particle production. So we're extremely excited about these new constructs, and we are in the process of testing them in vivo. And I just want to highlight that this work has really been driven by Dr. Kim Marie Dam in my lab, who is a scientific program lead, and she really leads these vaccine efforts. And she's probably in the process as we speak to screen the next construct. So hopefully there's more exciting stuff to come. In terms of characterization, of course, we're also very interested in studying hybrid mRNA vaccine-induced immune signatures. Like, how are these vaccines different in terms of the immune responses that they elicit compared to regular mRNA vaccines or standard virus-like particle-based vaccines? And so here we're really looking at things like, are there differences in germinal center activation? Are there differences in epitope mapping, T-cell responses, antibody effector functions? And of course, we want to understand where do these VOPs go in vivo when you encode them and deliver them as an mRNA vaccine. So there's a lot of work that is going on right now. And then, of course, we want to apply this technology to other vaccine problems, right? So as I mentioned, of course, we want to develop a pan-coronavirus vaccine. And of course, we're also interested in developing effective HIV, influenza, and malaria vaccines. And hopefully one day this technology could also be interesting for cancer vaccine development. So I want to spend the last few slides on our ongoing work on the universal coronavirus vaccine development. So just to remind you, the goal here is to elicit broad immunity to a wide range of coronavirus strains before human transmission has even occurred. And so the goal here is to develop vaccine strategies that target the immune responses to conserved epitopes. And so one of the leading strategies to target antibody responses to conserved epitopes is based on the development of mosaic virus-like particles. And so, this schematic just sort of talks you through the concept behind this idea. So if we're focusing on the left side, here we just have a virus-like particle that just presents a single antigen, okay? And so let's say this could be the SARS-CoV-2 spike protein. And you can see that the antigen on the surface is divided into these yellow and green regions. And so the yellow regions are the variable epitopes of the antigen. So this is the part of the virus that the virus can easily just mutate and where it can very easily escape from immune responses. And the green parts of the spike protein are the parts that are highly conserved. So these are parts that the virus really doesn't like to change, right? They're very important for the function of the antigen. And so what happens is if you have like a virus-like particle that only shows a spike protein from a single strain, you will mainly get strong activation of B cells that target the yellow variable epitopes because they are normally immunodominant. Like viruses are very good at hiding the conserved parts because they really don't want to get immune responses against these parts. However, if you then develop a mosaic virus-like particle where you're presenting, let's say, spike proteins from 8 different coronavirus strains, you will actually get weak activation of the B cells targeting the variable epitopes. And that is because B cell receptors on the surface of B cells are basically antibody molecules that have these 2 arms, so they basically look like a Y. And so to get good activation of the B cells, you really need both of the arms to be engaged. And so the problem here is that these B cell receptors that target the variable epitopes, they can only bind with one arm because the neighboring spike actually doesn't have these epitopes, right? Because it's a different spike protein. And so that then leads to preferential activation of B cells targeting conserved epitopes because the green part that is present in all of the different spike proteins is still there. And these B cell receptors can basically bind with both arms and therefore get activated stronger. And so this is the concept behind it. And my former mentor, Pamela Björkman at Caltech, and her postdoctoral scholar, Alex Cohen, have used this concept to develop a pan-sabecovirus vaccine candidate. And so they basically developed this mosaic nanoparticle that presents 8 different receptor binding domains from 8 different sabecovirus spike proteins. And they compared this here against a homotypic nanoparticle that only presents the receptor binding domain of the SARS-CoV-2 beta variant. And then they injected both of these vaccines into transgenic mice that have the human ACE2 receptor and basically then perform challenge studies. And so they either challenged with the SARS-CoV-2 beta strain, which was present on both of the particles, right? It was on the mosaic as well as the homotypic. And then they also challenged with SARS-1, which was not present on either of the particles, okay? And so then what you can see is that for SARS-CoV-2 beta challenges, both of the particles in blue and in red were completely protected, but then against the SARS-1 challenges, only the mosaic nanoparticle in blue protected, while all of the mice that received the homotypic particle died pretty quickly. And so this was a really nice proof of concept and shows this promise of this approach. But the problem is the way they are making these mosaic nanoparticles in the lab is actually extremely laborious. So, just to talk you through here, they're using the SpikeCatcher MI3 nanoparticle platform. And so in order to make these mosaic nanoparticles, they first have to express and purify all of these different 8 RBDs. And then in addition, they also have to make the SpikeCatcher MI3 nanoparticles. So they have to express and purify those. Then they have to mix them together. And then they have to purify the final product. So in the end, this is 9 expression steps and 10 purification steps to get this product, which sounds like a nightmare for any biotech or academic laboratory. And so this is kind of where I feel like our EABR technology could be, a really useful technology, because here you could simply just make the 8 RBD EABR construct, transfect them into the same culture, and the cells would just make the mosaic nanoparticles. And so we just wanted to see whether this would work. And so we performed this final experiments that I want to show you guys today, where we compared 3 different conditions. So on the left side, you just had homotypic. In this case, we picked SARS-1 spike. So SARS-1 spike EABR VLPs. Okay, so they only have SARS-1 spike. The middle group, which we call the admix, is basically a mixture of 6 homotypic VLPs. So they all just have one spike protein on the surface. It's 6 different spike proteins, and then we make the VLPs separately, mix them together, and then immunize the mice with that. And then on the right side, you have the final group, which actually made mosaic spike EABR VLPs, where all of the spikes are present on the same particle. Okay, and then we compared that. So we immunized mice with that, and then we measured the neutralizing antibody responses against SARS-1 on the left, which was present on all of the VLPs, and then against SARS-CoV-2, which was not present on any of the VLPs. Okay, and so what you can see is against SARS-1, all of the different conditions induced very high responses, and there were no statistically significant differences there. But then against SARS-CoV-2, you start seeing some real differences here. So the mosaic particle elicited over 10-fold higher neutralizing antibody responses than the homotypic particle. And the responses were also 2.5-fold higher than what we saw for the admix. So this really kind of like shows that the mosaic approach is really good and that we can use the EABR platform to develop these nanoparticles. But of course, what we really want to do is like see if we can deliver these mosaic nanoparticles through our hybrid mRNA vaccine approach. And this is what we're working on right now. Of course, there's some challenges here because you have to deliver 8 different constructs through an mRNA vaccine technology, and we're working on different ways to do that. And hopefully I can share the data about this exciting project with you guys maybe in the next session, hopefully soon. So this is all I have for today. So I want to, of course, thank all the people in my lab. We only started 5 months ago at the Gladstone Institute. I'm really thankful for all of them to be here, in particular, of course, Kim, who's leading this project. Of course, I also want to thank my former mentor, Pamela Björkman, and her group for their continued support and collaboration. I also want to thank the Caltech Merkin Institute for their support and our collaborators from Acutus Therapeutics, which are a leading company in the production of LNPs. And of course, I want to thank our funding sources from the NIH, the Gates Foundation, Wellcome Leap, the George Mason University Fast Grants, the Merkin Institute, and the Rothenberg Innovation Initiative. So with that, I'll leave it there, and I look forward to answering your questions.
Thank you, Dr. Hoffmann. That was a wonderful presentation. Let me quickly share our slides again. Yes. Yes, so with that, we will move on to the Q&A session. So anyone who has any questions, feel free to post them in the chat. And we already have a few questions collected during the talk. So first of all, are the VLPs that are synthesized in vivo, all the full VLPs are synthesized in vivo along with spike protein expression in the cells? This was a question from someone in the audience. Did you get the question, or do you want me to repeat it?
Could you please repeat it? Sorry. Yeah.
Yes. Yeah. Are the full VLPs synthesized in vivo along with the spike protein expression in the cells? Not sure yeah.
Sorry, I'm not sure
Yeah.
I understand the question, but like, so just to clarify, basically the in vivo, the mRNA will deliver a spike EABR construct. That spike EABR construct will then get translated inside the cells, and it will first go to the cell surface like the normal spike protein would do. There, it would then recruit the escort proteins from the cells inside the body, and that would then drive the production of these virus-like particles. So essentially, the spike protein would end up on the cell surface as well as on these VLPs. And so that's why we're arguing that this is combining the attributes of both a regular mRNA vaccine as well as a VLP-based vaccine. I hope that answers the question.
Yeah, sure. Yeah, if the person who asked the question, please let us know if you have follow-up questions for that. There was actually a second question from that attendee, which is, what are the chances of RNAi and nonsense protein production if mRNA is in the host cell?
Yeah, I mean, that's obviously a good question that applies to all mRNA vaccine approaches, not just ours. There has been some work on the nonsense production, and that's obviously something we need to look more into. It would also be interesting to see if that has different effects on our vaccine strategy as compared to a regular mRNA vaccine strategy. So yeah, these are things we need to look into, but yeah, it's not something we have looked at yet.
Yeah, there are a few more questions. So first of all, when you produce the EABR spikes on the VLP, do you think changing the amount of spikes on the VLP would affect the immune response? And have you try to titrate lower amount versus higher amount and then evaluated the result on the immune response there?
That's a really good question. So we To some degree, we have limited control over how many spike proteins actually end up on the VLP. It is kind of interesting. If you make these VLPs in vitro and look at them, you will rarely find a VLP that has less than 10 spikes on the surface. And I think part of the reason for that is you need a certain number of EABR peptides coming together to actually induce the process of virus-like particle production. I don't think just having one spike protein can induce the production of the VLPs. There's definitely you need to have multiple escort proteins being recruited to actually induce this process. So that's why it's difficult to make VLPs that don't have a lot of spikes on the surface with this version of the technology. We have now other, obviously, as we were screening so many different escort recruiting domains, we now have some constructs that have different properties. So they might make VLPs with fewer spikes on the surface. And so, but we haven't tested them in vivo yet. Now, the big question is this, is it more important to make a lot of VLPs, or is it more important to make fewer VLPs that have a lot of spike on the surface? And so this is really a question we need to answer. One thing to bear in mind is that when you deliver the mRNA in the LNPs, your expression overall is a lot lower than if you do a transfection with your DNA plasmid in culture. And we really need to do a lot more work to understand how these constructs behave when they're being expressed at low levels, right? And so I think it's very important to develop EABR constructs that can make virus-like particles very efficiently, even when they're expressed at low levels, because this is what we'll probably find in vivo. So yeah, there's still a lot more work to be done. My gut feeling would be if you have fewer spikes on the surface, that particle is probably going to be less immunogenic. That being said, there's probably some sort of threshold. If you have a certain number of spikes on the surface, if you further increase it from there, they're not becoming more immunogenic. So you just need to make sure that they're having that, that lower threshold of immunogens on the surface, and then they're probably great.
Thank you so much for that elaborate response. That was very clear. Yeah, we have another question. Do you anticipate that, so at some point in the presentation, you showed the data for day 50 something and day 112.
Yeah.
Do you anticipate that the immune response that you observe at day 112 would also last longer if the neutralization antibodies are higher at this point? Or is it not indicative of how long immunization would be ongoing? Or the immune response?
Right. I mean, so, I think I need to dissect this question a little bit. I think I'm 100% sure there is no more VLP production going on day 112,
Yeah.
right? Okay. And I mean, that wouldn't also happen with the regular mRNA vaccine. I think that the actual translation probably lasts a couple of days. I think it could be anywhere from 2 days to maybe a week. And where you get expression, it's probably more likely going to be closer to 2 days. That being said, there have been a lot of studies that have detected the protein, let's say the spike protein, after mRNA immunization, even weeks after the immunization. But I don't think that's a sign of ongoing production. I think this is just the spike protein is still there and it's still being retained. And of course, lymph tissues are very effective at retaining the antigen so that they can continue to train the immune cells to develop a more effective response. And we've seen this, not only for mRNA vaccines, but for many other vaccines. The germinal center responses can go on for months after the immunization. So they have strategies to retain the immunogens there in the germinal centers, and so your B cells will continue to get trained and develop more effective antibodies against the virus. But so the interesting thing with the durability is that you can think about durability in 2 different ways. You could either have, let's say, you get similar peak responses, but then one of the vaccines, the antibody titers wane less fast than for the other one, and then you get longer periods of protection. So this very much depends on your long-lived plasma cell population, which we haven't looked at yet. The other idea would be, and this was kind of more what we were promoting here, if you have higher peak titers, but they're decaying at a similar rate, you would still end up being, protected for a much longer time because you just started at a much higher level. And so these are still things that we really need to study in detail. And again, unfortunately, I don't think the mouse is the right model to do that. You probably have to go to non-human primates. And it is my understanding, I haven't done this experiment yet, but from other investigators, it sounds like that even that data might not translate that closely to what you would see in humans. So I guess in the end, you really kind of got to test it in humans. And of course, that's a real challenge, particularly when you're an academic lab. So I think there's still a lot to be explored, but I do think that durability is perhaps the most important thing to look at in terms of vaccine development going forward, because I think the situation that we had during the COVID-19 pandemic, where you need to get a new shot every 6 months, is just not ideal, right? Like from a cost perspective, but also from, just it's not convenient for people. I do think there's a lot of vaccine fatigue in the population right now, which is sad because in the end, vaccines are, probably the one biomedical technology that is actually working. It's like with everything else, yeah, we can't cure anything, we can treat things, but vaccines actually save lives. And so I think it's really important that we can get excitement in the population again about vaccines.
Great, thank you so much. That was a very clear explanation. We do have a few other questions from our audience. Yeah, so from an artificial intelligence, data science, and statistics side, do you see any possibility to contribute to this research?
Oh yeah, absolutely. No, that's a fabulous question. I mean, yeah, I mean there's a lot going on. I mean, I feel like AI has a huge potential for particularly the design of better immunogens. I mean, if we're particularly when we're talking about the universal coronavirus vaccine work that I described at the end of the presentation, if we can design immunogens that are better able to focus the immune responses to certain parts of the immunogen, that would be wonderful. If one could make minimal immunogens that basically get rid of the variable sites so that you only get responses to the conserved sites, that would be really, really wonderful. I think it's just very challenging to engineer these proteins sometimes to be that way. But I think with AI, there's so much going on right now, and I think it will just like open up the space to really design the best possible immunogen. So I think that's really important. Yeah, I think that's just one of the applications. I'm very excited about AI, and hopefully we can also use that particularly for our immunogen design part. And yeah.
Great, thank you. Yeah, then I think here is one more question. Are there challenges with bias in the expression of the delivered constructs?
Are there challenges? Sorry, can you read that to me again?
Yes. Are there challenges with bias in the expression of the delivered constructs?
Oh, I think that probably refers to the second part of the presentation on the mosaic VLPs. Absolutely. Yes, this is a huge problem, and this is probably, it's one of the challenges that we're facing. So let's say if you pick 6 to 8 different spike EABR constructs from different strains, some of them express really well and some of them don't express that great. And so it's really about finding constructs that kind of like all express well and all express similarly well. And then hopefully you can develop a well-balanced vaccine. We're working on some other strategies also to overcome that problem. But I think in the end, the big question is how balanced do they need to be? Again, if you had, like, of course, if you had 8 constructs and one of them would out express all the other ones, that would probably be bad, because then you pretty much really get a response against that spike. But let's say if you had 8 constructs and 5 of them expressed, let's say 40% better than the other 3, but you're probably already getting great diversity from just the 5 spike proteins. So you just have to test it and see if you're then suddenly not getting strong responses against one particular class of viruses. if but as long as you're getting good responses across the board, then it's probably okay. But yes, it is definitely a real challenge. And even for the bivalent mRNA vaccine approaches, like for instance, like I think Wuhan expresses definitely better than BA.5. And so, this is a challenge in doing this. But it's kind of funny because when Pfizer and Moderna made these vaccines, they just did it. I'm not sure how much they I don't know how much they cared about whether they expressed in a similar way. Obviously, it did help. You get stronger Omicron responses to some degree, even though for them it was a little different because they gave it as the fourth shot rather than the third shot. In our experiment, clearly as the third shot, you did see improved responses by adding that second antigen. So I assume that BA.5 expressed okay. Yeah, those are real challenges for sure.
Absolutely. Maybe there's time for one final question, really briefly. Have you, by any chance, evaluated the long-term effects of immunization in terms of gene or protein expression in other non-canonical immune cells and organs, such as, for example, heart or brain? Have you seen any changes there after immunization?
We have not done these experiments. That's super interesting, and I think that's super important. We absolutely need to do that. And I think you really look at this from two angles, right? Like first of all, you want to understand the immune response better, and so looking at different immune effector cell types and seeing how the gene expression profile is changed there, I think would be really important. I'm thinking particularly innate immune responses, I think would be super interesting. So this is certainly something we need to do. But then, of course, and I think this is also part of the question, we need to look at safety, right? So like looking in these other cell types like cardiomyocytes, etc. Hopefully you don't see any gene expression changes that would concern you. But of course, going forward, if we want to like develop this into a product, that's really, really important to do. And I think vaccine safety is absolutely paramount. It needs to, and I think we need to be better at like studying this early on and really in depth and understand better what these vaccines actually do, so that we can also develop the safest product.
Okay, thank you so much. That was great session. Yeah, then I would like to thank everyone for being here, and especially Dr. Magnus Hoffmann for the wonderful presentation and very insightful discussion. So with that, I would like to end and say that Creative Biolabs regularly invites distinguished experts in life sciences and also biotechnology to share their latest findings in our webinar series, as we have seen today. We encourage you all to visit our website and also follow our social media channels to stay updated on all our upcoming events. And that's it today. So thank you again, everyone, for joining. And again, thanks again, Dr. Magnus Hoffmann, for this wonderful presentation. And yeah, then we will see you all next time. Thank you.
Thank you so
And have
much
a
for
great day.
having me. All right, bye-bye.
Bye.